Good afternoon, class. Today, we're going to be discussing cell division. So if we will turn to our content objectives and language objectives, today students will be able to identify the parts of a cell during cell division. And then for our language objectives, students will be able to describe cell division and explain the differences between meiosis and mitosis. Now first we'll do a little review over what we covered yesterday when we did the introduction to cells. Can somebody tell me what a cell is? And it's perfectly okay to look at the word wall. Thank you for raising your hand, Sanja. Tell me what a cell is.
A cell is the smallest functional unit of life.
Very good, very good. And I'm glad you said functional because it's not the smallest unit, but it is the smallest functional unit. Okay. Now, another important vocabulary word that we learned yesterday was cell membrane. Now this one is a little bit more difficult. Can somebody tell me what a cell membrane does?
It makes up the outer layer of a cell.
Yes. And so by making up the outer layer of the cell, it gives the cell its shape and it holds everything in together. Mm-hmm. Okay. Now within that cell membrane is that goopy stuff we talked about yesterday. What is that called?
Cytoplasm.
Very good. Awesome job. I'm glad you guys remember. Okay. So within that cytoplasm is the brain of the cell. It does all the thinking for the cell. It controls everything that's going on with the cell. And that is called for $500?
Nucleus!
Alrighty. And $500 goes to Miss Ashley. The only other thing we need to cover again today are mitosis and meiosis because we're going to go in depth with those two. So does anybody know what meiosis does? Or what is the process of meiosis? Yes, Sanja.
It allows one diploid cell to divide into the haploid cells.
All right, so there's more than one haploid cell.
Yes.
Absolutely. All right, and mitosis?
Process by which a cell separates into two identical halves.
Awesome. Okay, now what we know is that meiosis is what is used in animal cells, and mitosis is what is used in plant cells. And so meiosis would be used in the human body, right?
Yes.
Okay, how many of you have ever known someone who was pregnant? Okay, everybody. Awesome. So what happens to a woman's stomach during the stages of pregnancy?
It gets bigger.
It does. It gets bigger. And do you know why it gets bigger?
Because the baby's growing.
Absolutely, because the baby's growing. And the baby is growing because the cells that are within the baby are undergoing meiosis or meiosis. And what happens is every day that baby is growing little by little. And so we can't see the growth on a daily basis, but from month to month you can literally see how much bigger a person's stomach gets when they're carrying a baby. Now today we're going to go in depth. We're going to talk about the different stages of meiosis and meiosis. And we're going to watch a little video. And we're going to make flip books. Okay?
Okay.
Now, flip books are what was used before we had computers to make cartoons. Okay, so before there was a Shrek, before there was a Monsters University or a Monsters Inc., we had to use flip books, which means that we had to draw each individual picture and change each one just a little bit so that it would look like the character was moving in the picture when we did the flip book. All right. So the first thing that we need to discuss is interphase. An interphase is the stage of the cell cycle when the nucleus is not dividing. Now I want you to pay special attention to the word interphase because if you look at the very beginning of the word, the suffix, the prefix, inter, is the same thing that you'll find in intermission, which means you take a break. The cells normally are in this stage for the longest time of their life because if they're not constantly dividing. They only divide for a short period of time.
Okay.
The next stage, or the first stage of mitosis or meiosis, is prophase. And this is when the nuclear membrane that's holding everything in begins to break down, and those chromosomes that live in the nucleus start to form chromosomes. Those strands of chromatin break down, well, they form chromosomes. Our next stage is going to be metaphase. So these chromosomes, they line up in the cell. Like we line up to go to lunch. They line up together so everybody's in order. Okay? And then with anaphase, they separate and they move to opposite ends of the nuclear spindle. And that's just like when we get in the lunchroom and you have your friends that you like to sit with and somebody else has their friends, and so everybody kind of goes their separate ways. It's the same thing. In telophase, this is the final stage. Because everybody has moved to opposite sides of the nucleus, there's a division that occurs, and so the nucleus of the new cells is formed in this stage. Finally, we have cytokinesis, which is where the cell cytoplasm is now divided. Okay, and so those are our first stages that we're going to go through. Now we're going to watch a short video that is going to describe the differences between meiosis, meiosis, and mitosis. Okay, and we're going to pull that up now.
Prior to meiosis or mitosis, DNA replication occurs. In both meiosis and mitosis, the nuclear membrane breaks down as the DNA organizes into chromosomes. In meiosis, chromosome pairs come together, or synapse, and crossing over occurs, resulting in mixing of the genetic information between the chromosome pairs. The paired chromosomes then align along the central plate of the cell and subsequently separate, one traveling to each end of the cell. In meiosis...
Okay, I want you guys to pay special attention here because that is the first difference between meiosis and mitosis. So I want you to take special note that in meiosis, the chromosomes actually cross over, and so it leaves a marker on each one. So you'll need to make sure when we do our flip books that you note that on your DNA, okay? On your chromosomes, rather.
A second division sequence occurs, resulting in 4 cells with half the number of chromosomes. Mitosis involves a single division sequence, resulting in 2 cells with no net change in the number of chromosomes.
Okay, that's going to be your second difference, because in meiosis, you're going to end up with 4 cells that all have different genetic markers. Whereas in mitosis, you're going to end up with 2 cells that have the exact same genetic makeup. They have the exact same number of chromosomes. Okay, so remember that was the diploid and the haploid? Right, okay. And that is the end of our video. That was short and sweet, wasn't it?
Yes, ma'am.
Yes, it was.
I didn't want to bore you guys with a whole bunch. You're so very welcome. Okay, so now let's turn to our flip books. This one is the meiosis flip book. And so you're going to go through, and in each slide, you're going to draw a picture of your cell. And you're going to draw what happens in each stage of division, okay? Now, you can also refer to your textbook, because I know that video went by really, really fast, okay? So you can refer to your textbook, and I'm going to allow you to work in groups to get this done, all right? Now, we'll come to each group and make sure that everybody's on task. And if you have any questions or anything, you can definitely ask me, okay? So like I said, you'll draw each picture, and in each picture, you're going to have to show the changes that happen as you go from each stage to the next, okay? Now, the other thing that's very important, I'm going to show you one that I've already done. And Miss Thomas was in a hurry, so it's not neat and even, okay? And that is very important, because you want to be able to flip your book and see the cell change and go from 1 cell to 2 cells, or 1 cell to 4 cells. So on each page, like I said, you're going to have to draw and label your cell and the different parts, and color in your DNA, well, it's going to start out as chromatin material, and then it's going to become chromosomes. Okay, and like I said, on each one you're going to have to label what is happening in the cell. All right, and so that way when you're done, you'll be able to flip it. But remember, you need to make nice, even scissor cuts so that our books will turn out right, so that they don't end up being sloppy like Miss Thomas's.
Okay.
All right. Okay. So we're going to take about 15 minutes to do the first one, and then we'll stop and we'll talk again about meals. All right. So what we're going to do is we're going to review what we've already covered today, which are the stages of cell division. And the stages are IP-MAT. So the first stage, which is interphase, that's where our I comes from. What is happening during interphase?
It's at rest. It's resting.
Right, the nucleus is not dividing. So remember, like intermission, when you take a break and you're at rest. All right, so our second stage, or actually the first stage of mitosis or meiosis, is when the nuclear membrane breaks down. What stage is that?
Prophase.
Prophase. That would be our P from IP-MAT. Now, the other thing that happens during prophase is that the nuclear membrane breaks down, but the strands of chromatin form into chromosomes. Remember that as well. All right, so next is a stage where we meet in the middle. So that would be
metaphase.
Metaphase, exactly. So meet in the middle, metaphase. And so that's where the chromosomes are going to line up along the equator of the spindle. All right. Now, in the next phase, the chromosomes don't like each other as much. And that stage will be
anaphase.
Anaphase. That's where your chromosomes move to opposite ends on the nuclear spindle. Now, the last stage of mitosis is telophase. And this is what happens to the daughter cells in telophase.
They group into the new nuclei.
Right. So the new nuclei are formed, and they are two totally separate cells now in telophase. And so in cytokinesis, the cell membrane is also going to be separated, so you have two completely separate cells.
Okay.
Because in telophase, the nuclei are separate, but in cytokinesis, that's when you form two totally separate cells. I want to make sure we're clear on that. All right, now in meiosis, we're going to have a second set of stages, okay? So you're going to go through prophase, metaphase, anaphase, and telophase all over again, all right? So in the second prophase, your nuclear membrane is going to break down, the spindle is going to form, and they're still going to meet in the middle in metaphase, and the chromosomes are going to line up on the equator and attach to the spindle. Now that is a little different too from the first metaphase, because even though they line up on the equatorial plate, they're not attached to the spindles either, all right? Now in anaphase, the chromosomes are going to separate, and they're going to sit on opposite sides of the lunchroom, just like you guys do when you group off with your friends. And then the last stage again, telophase 2, is when the 4 daughter cells are produced, and they're going to have how many sets of chromosomes? 1 set of chromosomes. Okay. Now, will those chromosomes be identical? No. Very good. Awesome. You guys remembered. Okay. So now we're going to do our meiosis flip books. We're going to take a 15-minute time to work in our groups on our meiosis flip books. You guys did an awesome job making sure that your cuts were even so that they flip right. So I want you guys to do the same thing. I want you to pay attention to detail and make sure that that happens, okay? So we're going to take 15 minutes for that. I'm going to walk around to your group, so if you have any questions about anything that we've covered, you can always just let Miss Thomas know and I will answer your questions, okay?